So, okay. So now it was. So I'm interested about the RNA molecule. That is a molecule that has play several crucial roles in the cell. There is one that is a messenger RNA that has the role to bring the DNA information and to produce a protein. And yes, soon it was working. Sorry. Very sorry for that. So, I'm interested about the non-cloning RNA that is folding, and the folding of the RNA is very important because it is associated to the activity of the RNA. So here in our study, we are more interested about the RNA secondary structure. So here is a schematic scheme of what is a non-cloning RNA structure, where we have different structural elements, which are the external loop, the bulge loop, the hairpin loop, the stem, and so the different other structural elements. Here in our method, we are more interested about the structural element, except the G4, it is more tertiary tertiary structure, and the columns that make more difficult to compute the evolution of RNA structure. So in the computational context, there are many methods that have been developed for non-cloning RNA structure for the RNA structure prediction, and also to build accurate RNA multiple and sequence structure alignment. And this allows us to infer evolutionary history of the structure of RNA families, but until now, very few number of methods have been developed for such a problem. Here is one of the methods that have been developed for such a problem, that is an indigram. So it's a method that takes as input a phylogenetic tree relating several structured RNAs, a description of the evolution of structured RNA along a single branch of the tree, that is in the form of a pair stochastic context free grammar with a model that is a TKF91 structure tree, that is a model for RNA structure. And so the model is has a possibility to reconstruct evolutionary of the RNA structure with deletion, insertion in the different loop of the sequence and loop of sequence stem sequence. And at the end, it will output a phylogenetic multiple sequence grammar, and a posteriori will make a reconstruction of the evolutionary history of the RNA using this grammar. The limitation of this method is not applicable on a large data set, and there was not an implementation available for this method. Another one is the Ashalman. So it takes as input two non-cloning RNA families that share a theoretical common ancestor with a single copy gene. So here is an example where you have the two families in red and in blue. So the two families have to share the same species and just have one single copy gene for each. And there is only one consensus structure for each family. And after, with a method that is based on Fitch parsimony method, it will reconstruct the ancestral sequence based on the comparison of the consensus structures. That is that when you go on the root of the tree, you have both of the consensus structures that will have the same weight to reconstruct the ancestral structure. So the limitation is there is only infer ancestral sequence and not the structure, and also there is not the indel are not allowed. There is only consensus structure, but not a structure for each sequence of the method. So here in how research progress, we have the goal to develop a new algorithm to infer ancestral RNA structure by solving the Robinson-Foulds small parsimony problem on phylogenies of RNA trees. So here I will represent you the different parts. So first we come with the RNA multiple element, where here we have 4 RNA, A, B, C, D, where we have the sequence of the RNA and also the lines that represent the structure. It's a dot bracket format, where we have a parenthesis, in parenthesis, and out parenthesis that gives a pairing between the sequence, and so it gives you the folding of the RNA structure. So here first we have the goal to make some block delimitation to make a comparable block between the different RNA sequence and structure of the sequence. So the idea is to see the pairing status for each sequence in the alignment, and when there are continuous status all along the sequence, we put it in the same block, and when we have a change, so for example here we have a gap, so we have a change in the status of the pairing, we add a block delimitation and we go up on the sequence. So at this position we see we have a gap, and just after we have in parentheses, so we have to define a new block, and we go until the end of the alignment, and we have the block delimitation for our multiple RNA alignment. After that, we also have to check for the different stem structure, because as we see here for each sequence, the out parentheses are continuous here, but we see that the first 4 parentheses belong to one stem, and the third after belong to another stem. So here we have also to add a new block delimitation to because we have a block has to be associated to only one structural element. So here after it is the definition of the setting of the block. After that, the idea is to take this sequence structure element and the block delimitation, and to represent this sequence structure of the RNA as an RNA tree, where we have internal nodes that represent the structural element of the RNA structure. So here we have the S for stem, I for internal loop, B for bulk loop, and H for hairpin. And after, to each of these structural elements, we associate the block that are associated to the structural element. And we make this so for all of the RNA structural elements. And also we take as in input of our method a phylogenetic relationship between the two between the different RNA trees to get a phylogeny of RNA trees. After that, there is in our algorithm we have to enumerate the list of clades of the RNA. So we take the RNA tree and we enumerate all the clades from the structure. So here's the enumeration of the clades for the RNA A. And after that, we have the phylogeny of RNA trees with the list of clades. So here, the problem we are interested is the Robinson-Foulds small parsimony. So in our problem, the Robinson-Foulds metric is the size of symmetric distance between 2 RNA trees. And the Robinson-Foulds small parsimony problem is as input phylogeny on RNA trees, I showed on the previous slide. And after it will output a set of RNA trees at the ancestral node that minimize the sum of the Robinson-Foulds distance on edges of the phylogeny. So after we have also clades on the ancestral node that will be interpreted as RNA trees. So here's the method as it works. So it will compute the first time the putative set of clades on ancestral node. So the putative clade that consists to the set of all clades that are present at least in 2 edges at each node. After that, for each node, we get the clade of the weight of each clade. The weight of a clade, so that is belong to a node, is to take for each if it's present in the edge at the intersection of 2 edges of so the intersection from this side, the intersection from the child, and the intersection of from the parent and the child left. And after that, we compute the maximum weight set of compatible clades. And after we recompute the optimal median at each ancestral node recursively across the phylogeny until we reach a stability about the optimal median. So here I represent some results. So I will present a case study that is for the RNA family RF04164. It's from the dataset from RFAM database in the release 14.7. So for this family, the element length was of 108 nucleotides, and it contains 8 RNA sequences from 5 species. So here is represented the species tree, and here is the RNA family tree for this. And so that is the result we get with our method. So here is represented, so for you have each leaf represents the RNA, and after the numbers will represent the ID for the different clades that have been associated to each of that are present in the list here, and after you have the list of clades for the ancestral node. So here there is a color code. It's because we have the clades that are with the color purple represent the clades that are the common shell structure between all the sequence. We all see that they have at least one of this. So 68 is the most basic structure that it's composed of the different. The block is the notation is in Roman number. That is represented here, and after we to avoid to make too complex to the presentation of the block, I add the clade ID that represent a set of clades. So here we see that this clades are shared, are present in the ancestral node, and why I put some color. So in green, it represents the clade where there is no missing block, because in this one, when you have in your multiple alignment gap, most of the time you have the block of the gap is missing because there is no sequence associated in the structure. So sometimes the gap the gap means that you change the type of the number of your clade. So here is a missing block. Is in red is represented the clade where there is missing block, that is a 23 block that is not present. So if we compare here, we see that there is a 22 and 24 identity block, and after, so there is a missing identity block in this clade. That's why they share after all clades. The 79 is included in the 50, that is included in 46, etc. And after we have also this the blue one that represents a missing block for the 23 to the 25 missing block. So here, as we represented the evolutionary of RNA tree, we see that it infers so the basic structure that is shared by all the RNA structure, and after it will infer so more the clades that don't contain this block. So our method will infer the absence of this part of the sequence and structure at the ancestral state, and after that you have the insertion of this block. So first in the red one, so you don't have also the 23, but in the green one, you have you see the insertion of the 23. So in conclusion, so we defined the new block delimitation of RNA multiple sequence structure element to study the evolution of non-coding RNA structure. This is a new computational method to infer ancestral RNA tree clades with block delimitation based on a heuristic solution of the Robinson-Foulds parsimony. And there is a presentation of a case study for a proof of concept with a heuristic solution of Robinson-Foulds parsimony. In perspective, we'd like to apply our method on the complete airframe database. We also need for this to finish implementation of the method, because for now there is not a statistical analysis of the ancestral RNA trees inferred by the method. And we also need to create a Git repository for free availability of the method. There will be also in project the development of a method for the Robinson-Foulds for large parsimony problem to infer the RNA family tree and compare it to other phylogenetic tree for the RNA family. And also there is actually a related work in progress in the Cobius lab with Bertrand Marchand, Manuel Lafont, and Aida Gombera, that is to explore the theoretical complexity of the Robinson-Foulds parsimony problem, and also explore other distance metrics such as the tree-edit distance, and also extend to other problems such as the median problem. So I would like to thank all the members of my lab, the Cobius lab. Also, I would like to thank the organizer for giving me the opportunity to present this talk, and also the collaborator, Bertrand Marchand and Manuel Lafont, for the collaboration on this project. Thank you.
Okay, thank you. Is there a question?
I don't get the microphone.
So it's late in the afternoon and maybe I missed something, but I'm having trouble figuring out what you're trying to reconstruct. So I usually think of structures as being a phenotype rather than a genotype, and so how can you have a tree for that? Or are you using it as a marker for understanding the history of something else?
For the phylogenetic of the RNA family, you want to say? So this, because the idea is you have as input the phylogeny, so the phylogenetic inference for the RNA family, and after you have the RNA tree at each of your leaves. And the idea is also to reconstruct what is the non-conic RNA on the ancestral state. So it's to compute the ancestral RNA trees.
So you're doing ancestral state reconstruction?
Yeah.
Okay.
Ancestral state.
Oh, okay. So I'm with you. Thank you.
Okay.